Good morning, everybody. Welcome to our Gene Delivery Seminar series for the month of April. I'm very glad that you're all here. I am very pleased to introduce today's speaker, Dr. Francis Enane. He is a scientific investigator and biotechnology leader with experience spanning biopharma, biotech, and academic research. Dr. Enane earned his PhD in molecular medicine from Cleveland Clinic Lerner College of Medicine. He currently serves as senior staff scientist at Indiana Biosciences Research Institute, where he leads assay development, automation, and translational research efforts across oncology, rare disease, and cardiometabolic disorders. Previously, he held scientific leadership roles at Novo Nordisk and Beckman Coulter Life Sciences, advancing drug discovery, high throughput automation, and precision medicine. Today, he will speak on designing transient mRNA therapeutics for next wave medicine. We are very delighted to have you with us, Dr. Enane. You are welcome to start whenever you feel ready. But before that, I do want to mention that we will be taking questions. If you have questions, there should be a little Q&A box at the bottom. So please type it in there and we will get to it towards the end. Thank you. And this is being recorded. And with that, whenever you're ready, take it away.
All right. Thank you, Samantha, for the introduction, and thank you everyone for joining the meeting today. Obviously, I'm extremely excited to be able to share this very emerging topic with you, where we'll talk a little bit about the biology of the mRNA and how we can convert the mRNA into a therapeutic product. So we have a bit of a loaded agenda, but we'll try to get through it together. We'll start by talking about the rise of the transient mRNA medicine, and then we'll go into the details of how to engineer mRNA into a potential therapeutic, how we deliver it via different vehicles, and then optimizing this particular mRNA for tissue-specific targeting, and then we'll wrap it up by talking a little bit about the translational case studies and how the mRNA therapies are now moving beyond the liver as a tissue of interest. So the mRNA is a specific molecule that allows precise transient protein expression. In the context of disease therapeutic development, it is ideal for vaccines, development of immunotherapies, as well as development of transient protein replacement strategies. The clinical momentum for this type of therapeutic approach was really strengthened through the COVID-19 vaccines, which has now grown across different pipeline, including oncology, infectious diseases, and most recently rare diseases. So the platforms are currently moving beyond vaccines, and we are getting into this precision programmable therapeutics, including self-amplifying RNAs and circRNA, as well as developing cell-specific constructs for therapeutic effects. So the evolution of the mRNA technology can now be grouped into 4 different categories. In the 1960s, between 1960s and 1984, we were really in a discovery stage. And in 1961, there's a publication of a paper by Brenner and Melson, where they basically described this unstable intermediate molecule that they saw it as a carrier of information from genes to the ribosome for protein synthesis. And so they call this molecule messenger RNA, which is what we abbreviate as mRNA. So it was the beginning of the identification of the fact that we can go from a DNA to a protein through a very cell-specific synthesis process. During this period, we started to then learn a lot more information about this particular molecule, where we were able to discover cell-free translation systems. And those studies were able to confirm that it is indeed an mRNA molecule that can produce the protein. But the other thing that we knew at this time is that the mRNA structure was generally very unstable. And so we started to do research to look more into how do we stabilize the molecule which led to various discoveries, including the 5' capping structures that allowed the stability of the molecule for us to now think about path to synthetic mRNA production. And then there's another discovery of the T7 or SP6 RNA polymerases. And what those discoveries did was to enable the capacity to be able to scale in vitro transcription of mRNA to a level that we can then start to move to the next phase, which is the platform phase. And the platform development started in around 2000, where different studies were trying to establish how to modify RNA to enhance its stability for in vivo applications. And a landmark study was by Carico and Wiseman, and these two won a Nobel Prize in Medicine recently for their work. But what they were able to demonstrate very effectively was that you could not only synthesize mRNA in vitro, but you could take that mRNA that you've developed in an in vitro system and inject it into an animal and allow it to produce protein of interest. So at this time, we started to look into therapeutic effectiveness post mRNA injection and whether it was feasible to be able to develop a therapeutic using an in vitro design messenger RNA. So some of the important findings and discoveries in this era was the nucleotide modifications that are necessary to reduce innate immune reaction against an mRNA molecule that allows for ability to develop safe and repeat dosing strategy. They also, we started to understand method of delivery where we used a lipid nanoparticle as strategies for packaging the mRNA into the lipids that could enhance the mRNA uptake while protecting the mRNA from potential degradation. And then we started to learn about codon optimization, engineering the UTRs for specific modifications, and then strategies to purify the produced molecules so that they are safe and can be injected into animal and human use. So that propelled us into the clinical translation piece, where we could then start to see, is there a route for clinical application using mRNA therapy? So around 2010, all the way to around 2013, there was a lot of work focusing in developing cancer vaccines, where you could personalize mRNA such that it can take advantage of the genetic mutation on cancer cells and develop a personal vaccine where the immune cells can attack the mutations present on cancer cells and essentially eliminate the cancer from the body. There was some work ongoing in developing mRNA vaccines used against rabies. And then the memorable time in the use of mRNA was in 2020, where we developed this COVID vaccine against the COVID-19 virus, which was heavily pushed through Moderna and BioNTech vaccines. This basically led to effective validation of the safety of mRNA molecule, but also the validation of the vehicles of delivery of mRNA therapeutics. So basically, millions and millions of people were injected with the COVID vaccine, and that provided a lot of heavy and useful data that can demonstrate that the mRNA as a therapy itself is safe, but also that the vehicles that we use to deliver that mRNA are also safe for human usage. So there is currently a lot of clinical validation of different classes of mRNA therapeutics. Some are looking into cardiovascular diseases and they're expanding into non-vaccine-based therapeutics such as protein replacement, which is now leading us into the final phase here, which is the commercialization stage, where we have multiple different groups trying to develop therapeutics across different types of diseases, including rare genetic diseases, oncology, cardiovascular, infectious diseases. So the applications for mRNA therapeutics are extremely wide. And there's a lot of R&D work ongoing to effectively establish the ability to use these molecules in therapy. So how do we optimize the mRNA structure to get it to a clinical stage molecule? So I'm going to talk a little bit in detail about all this, but really your naked mRNA is a very unstable molecule. And so a lot of efforts is to try and to stabilize it. And there's different levels of stabilization that one can decide to do. And we're going to cover some of them in detail. You can also, depending on the disease that you're studying, if you can use different classes of mRNA, you can use the single-stranded mRNA, or you can use the self-amplifying RNA, as well as the circular RNA. Obviously, these are going to be context dependent in terms of your goal. But once you've selected the structure that you want to use and perform the specific modifications necessary to improve stability, you then have to think about how am I going to deliver it into a patient. So there's multiple different vehicles that can be validated and evaluated for application of interest. I'm going to talk a lot more about lipid nanoparticles, but there are other vehicles of delivery that one can validate depending on what they're trying to accomplish with the mRNA. Obviously, as you go through solutions, you also go through challenges. And one key challenge that one has to think about is if you are delivering your mRNA depending on a specific delivery vehicle, are there any off-target things that you have to think about? And so those are things you have to pay attention to as you decide different modifications as well as different delivery methods. And then in terms of applications, I think the applications are quite wide. People are developing epidemic vaccines, cytokine therapies, cancer vaccines using neoantigen technology. You can develop cell therapies as well as protein replacement therapies and even gene editing, which we're going to be talking a little bit about in the next few slides. So really, to develop a mRNA therapeutic, there's an integrated platform, and this integrated platform really involves the chemistry that goes into synthesizing a specific molecule, which delivery route are we going to use, and then using biology to integrate the data that you are generating and essentially go through this loop of chemistry modification, delivery, and data feedback. So obviously today it's always hard to not incorporate artificial intelligence as an option to help you design, optimize, screen, and then use data as a feedback to then continue to modify. But really what you need to think through is the design strategy for your codon sequence, which modifications you're going to use, formulation, delivery, and then the therapeutic intent. Are you able to accomplish what you're trying to accomplish with your mRNA development? So how do we engineer these molecules? As many of you know, mRNA is typically transcribed from the 5' to 3' end. And what we do is you take advantage of the knowledge that we have about these different pieces of the mRNA, whether it's the 5' end, the coding sequence at 3' end, or the poly A end of your molecule. And then you modify specific structures across each section of the mRNA with the goal of improving stability, enhancing expression ability, and maybe observing or tracing where your mRNA is going. So at the 5' end, you typically want to cap the 5' end because there are specific sequences at the 5' end that are prone to endonuclease degradation. And those endonucleases are what's going to limit how long your mRNA is expressed. So if you can cap the 5' end with different capping structures, most people use these phosphodiester structures like the one seen in panel B over here. What that does to your mRNA is that it increases translational efficiency and also improves the lifespan of the mRNA, avoiding also the innate immune activation. For the body of your mRNA, what you have to pay attention to is specific nucleosides that can be modifiable without affecting the expression kinetics of the mRNA of interest. So what we've started to learn is that replacing certain sequences, such as uridine sequences, can allow efficient mRNA expression and also suppress immune detectability of the mRNA of interest. And then at the poly A tail, what you want to also do is to exclude sequences that can be detected using exonuclease enzymes. You can conjugate specific reporters to this end, which can help you trace where your mRNA is going, how long it's being expressed, and basically these modifications can also improve the lifespan of your mRNA and potentially improve your translational efficiency. Some people are doing fancy stuff. They can add chimeric molecules at the poly A tail. Obviously, those will increase diversity of what you're trying to accomplish synthetically with a specific therapeutic intent. So, these are different moieties that you can use to perform nucleoside modifications, and then different bases that can be essentially modified for specific therapeutic goals. So really a quick summary here is that at the 5' end, you are focusing on different capping structures that can enhance ribosome recruitment while decreasing immune activation, as well as enhancing the translationability. On your coding sequence, you're trying to establish which codons can be modified. You want to focus on your GC balance and which type of modifications are going to enhance your mRNA stability. And then at the 3' end, you are looking at modifications that can improve your mRNA half-life. Poly A tail modifications are going to help tune your mRNA to increase stability while enhancing translation. The other thing to keep in mind is what type of mRNA are you designing? Because each mRNA is not created equal. If you are designing an mRNA vaccine, your goal would be to essentially produce very low amount of protein, but that protein should be sufficient to enhance immunogenicity. So in this context, we want the immune activation to act against the protein or the mRNA that you are developing. So here you want to think about what your carrier is going to do. Obviously, we don't worry so much about a tissue tropism, as long as if you deliver it in vivo, you're going to allow immunogenic activation. So local delivery is efficient for vaccines. A low mRNA dosage is sufficient to increase immune activation, and these tend to have low toxicity. Your pharmacokinetic intent for a vaccine is going to be obviously low protein threshold, but effective amplified immune activity, which can react against your mRNA and elicit an immune response to the antigen that you're trying to target. So obviously clinical target-wise here is to enhance immune memory so that a specific antigen can be easily detected by the immune system, and immune protection can essentially be produced by your mRNA therapy. When it comes to the mRNA therapeutic, the goal is a little bit different here. So for mRNA therapies, what we want to do is we want to elicit a lot of protein. We want to produce a lot of protein, but we want to minimize immune activation because we don't want the immune system to eliminate our mRNA, because we want that mRNA to be produced to produce a protein of interest and then to elicit a biological process. So here we have to think about tissue tropism. We want to ensure that our protein is going to go to the tissue of interest and produce the biological activity. So we have to essentially determine which in vivo delivery route are we going to use. Most delivery routes, especially if you're using lipid nanoparticles, they're going to go to the liver. So how are you going to change your mRNA or the lipid particles to ensure that they can bypass the liver if the disease that you're trying to treat is outside of the liver? The pharmacokinetics obviously are going to be dependent on high protein yield, limited duration of activity, chronic delivery of your protein of interest, and then the ability to be able to repress the immune activation. The clinical targets obviously can vary. One, you can look at enzyme replacement. You can look at treatment of solid tissue cancers, tissue penetration, tissue regeneration is a effect of your protein of interest. Obviously, the clinical target is going to vary depending on the disease and what you're trying to accomplish therapeutically. So I wanted to a little bit emphasize on this immune response issue. So this is a study where they are trying to elicit an immune reaction, and they are trying to do that by evaluating modified versus unmodified mRNA to see whether they produce the same activity. And so what they do here, they injected rhesus monkeys with an unmodified mRNA, and then do two different doses of modified RNA. They will pull blood out of these monkeys with the effect of immunophenotyping and trying to identify specific immune cell markers that are activated. And then they want to monitor across different injections and different classes of mRNA treatments, which type of immune activation is happening. So I know this is hard to see, but what the summary demonstrates is that if they inject mice with modified mRNA, they are getting different type of immune cell activation. And if they inject, sorry, these are not mice, but monkeys. And if they inject the monkeys with unmodified mRNA, they are also getting a different type of immune activation. But the mRNA is the same. They're not changing the mRNA. The main difference is that this mRNA has specific modifications on the sequences, and whereas this mRNA is just the naked mRNA that is modified. However, both of them elicit an immune response against the antigen of interest. So the difference here is that depending on what you want to accomplish, you may need to use modified or unmodified mRNA, especially if they're eliciting the response that you want. Obviously, the modified mRNA might last a little bit longer, may have better memory. Who knows what it does, but it will accomplish different things. So as you think through whether you want to modify or unmodify, one goal is to determine what do I want to activate. If there's specific immune markers that you are trying to look for, you can decide which specific mRNA can be of interest to you. But this is in the context of a vaccine. When it comes to a protein replacement, the goal is quite different, right? So I'm going to illustrate that using this study that is looking at cystic fibrosis. So if you don't know cystic fibrosis, this is a disease that is complicated through a genetic mutation in a gene called CFTR. Patients that have these mutations, they have inability to transport chlorine efficiently. And so this disruption of chloride transportation leads to accumulation of thick mucus in the lungs and in the digestive system. And this can be a devastating, deadly disease if it's untreated. So one way to treat that disease essentially would be to design an mRNA strategy that can produce the CFTR, the gene product of the CFTR gene to effectively reverse this disruption and allow for patients to not have this thick mucus accumulation in their body. So here you can see two negative controls, and then they are trying to evaluate four different things. One, can they put DNA coding for the CFTR and be able to produce protein of interest? They can see that. And two, they inject a naked mRNA for CFTR and see if they can produce the protein of interest, which they also see that. And then here they have two different modifications on the mRNA, and they're also trying to monitor if they can produce the protein, which they do see that. Now, the difference is how long does the effect of what they're doing last? And then also, does it elicit immunity? So here you can see if they treat the cells with the DNA only coding for the CFTR gene, they're able to produce a substantial amount of the product. If they inject just the naked mRNA, they produce the product, but not in large quantities. Whereas if they have the modified mRNA and the two different modifications, they're able to yield a higher level of the product. If they look at the different classes in two different systems, you can see again at baseline the three different classes of injections are yielding the product of interest. However, the naked mRNA yields the least amounts, whereas the modified mRNAs are producing a higher level of the product. But most important is, what do the modifications do? So remember, the goal here is we don't want to enhance immune reaction. We want to reduce immune reaction. And so here you can see that they have different things going on here. There's the blood. They suck the blood, and then they look at the effect of the nanoparticle. There's obviously a positive control over here, and then effects of the different things that they're putting on here. So if they just put the naked DNA, there's a little bit of immune reaction, activation of the immune cells. You can see that the gray is linked to activation of interferon gamma, interleukin 8, and TNF alpha. However, if you look at the naked mRNA, naked mRNA produces a little bit of immunity, but it varies depending on the different classes of immune cells. Whereas the modified mRNA here, they do not produce this similar immune activation. So obviously this is important because it suggests that to be able to suppress immune activation against your mRNA, you might need to modify the mRNA in the context of protein replacement therapeutic development. So we'll talk a little bit about mRNA synthesis and the role of lipid nanoparticle as vehicles for delivery of the mRNA. So the evolution of in vitro transcription has been the most efficient route for scaling the mRNA synthesis. And what you need is you're going to need a template. So where's the source of your mRNA going to come from? So here you have a plasmid DNA probably with the T7 promoter, and then you use design softwares to be able to synthesize the specific sequences that can generate the linearized template for potential in vitro transcription of the mRNA. At this in vitro transcription stage, you start to think about the specific modifications that can be performed on the coding sequence. What type of capping structures are you going to use? And then which types of purifications are you going to then use to clean up your product? Think about the quality control system that it has to undergo to remove impurities, and then how you're going to formulate that. Now, all these processes are obviously automation friendly. If you are a person interested in doing things in a more hands-free environment, many of these processes can be automated on different liquid handlers and as a way to eliminate potential human error. And then obviously, as you synthesize your product, you have to think about methods of purification. I think if you're in your early research stage, you can get away with certain cheaper, low-cost methods such as lithium chloride purification. The goal of purification is to eliminate impurities, to remove double-stranded RNAs or truncated RNAs, salt that is left during the carryover stages. And depending on what you're trying to accomplish, obviously, you have to think through which method of purification you're going to use. Obviously, the most reliable, expensive, so HPLCs or magnetic beads that are frequently used here, and those are essential for preclinical or clinical stage type of molecules that are being developed. And then in the context of the mRNA modalities and the expression kinetics, I think the conventional mRNA can be used, but what you have to keep in mind is that you are going to basically have your target expressed very quickly, but it's going to have a short duration. And if that's your intent, you certainly can use it, but really the effect is not going to be lasting. If you modify your mRNA, you're going to enhance translation, and then you're going to have it expressed moderately. And so the duration of time is a little bit longer than the conventional mRNA. If you want a delayed onset for your mRNA, then you can think about self-amplifying RNAs. This will have delayed onset. However, the duration is going to be longer. The same applies for the circRNA. These ones have slow onset, but the durability is very stable. The other things that people are validating is this what we call trans-amplifying RNA, and these are kind of tunable or tunable. You can use specific things to kind of turn them on and off. So obviously, which type of mRNA you use is going to be dependent on your therapeutic goals and how you're trying to especially deliver that particular mRNA. But that being said, different modalities may use different types of mRNAs. What we've seen is that single-stranded mRNA are very good for vaccine development. They are obviously can still undergo different modifications of interest. The expression kinetics is that they are rapid and they can last from hours to days. These are what we used in COVID-19 vaccines by Moderna and BioNTech. A lot of the personalized cancer vaccines have been using these single-stranded mRNAs. And then obviously the primary application is going to be immunogenic activation, cytokine expression. In the context of cancer, people are engaging with these CAR T cells. The self-amplifying RNAs are obviously replicase dependent. And so these are used in the context where you need a long-lasting expression profile. This can be used for pandemic preparedness, obviously, vaccines, oncology, and the dose-sparing vaccine or immunotherapy are the primary applications. circRNAs, obviously, the circRNA it relies on covalent closed-loop RNA. They are highly stable with a very prolonged translation. Most of these are in preclinical early stage of validation. Potential areas of application is in situations where there's chronic protein replacement need, but also for vaccine where you need the molecule to stay for longer period to be ready to expose a specific antigen and allow that antigen to be essentially eliminated through the immune system. And then there are other classes of mRNAs that people look into. So microRNAs are essentially of interest for different diseases. Obviously, delivering this extrahepatic is a challenge, but there's some efforts where there's some improvements to deliver extrahepatic mRNA in this context. So next we're going to talk a little bit about method of delivery by focusing in on lipid nanoparticles. So liposomes were discovered in 1960s, but they really came into clinical utility in the 1950s. And so the discovery really was the identification of this hydrophilic and hydrophobic containing structure, where the phospholipid, where they mimicked were the phospholipid. The phospholipid basically has this hydrophilic tail or hydrophilic head and the hydrophobic tail when in aqueous solution. So the liposome basically contained the same structure. And so in 1995, there's a first use of a liposome to deliver a drug called Doxil, which is just a chemotherapy drug approved by FDA in 1995. But they essentially packaged this drug into a liposome, and then they were able to deliver a chemotherapy by this method. And then over a period of time, there were evolution of lipid nanoparticles. We started to learn more about ionizable lipids. which allowed endosomal escape for different cargo. And in the recent time, we've learned a lot more about different classes of these lipids. We have the pegylated lipid, which allow control circulation time. Cholesterol lipids, which allow stability as well as fusion to the plasma membrane. And then we have the ionizable lipid, which allows for cargo delivery into the cytosol to be able to produce a specific protein into cells. So the first drug used in this context was Onpratto, which is approved saRNA therapy that can be used for treatment of a disease within the liver. And for a long period of time, as long as your disease was in the liver, you could deliver your mRNA in that specific liver. And then recently, this has started to change a little bit, where we've started to design lipids that can go beyond the liver. And so the first one obviously was the COVID-19 vaccine, which was an intramuscular injection that effectively delivered a systemic vaccine that protected many people against the COVID pandemic virus. But then from that, they started to emerge what we call the on-target lipid composition. And so people have been trying to modify the different lipids to try and see if they can push delivery outside of the liver. And so we'll talk a little bit about these types of modification in a bit more detail in a couple of slides. But the way you obviously handle these lipids is via what we call microfluidics. So when you take your lipid composition, you want to combine it in such a way that it creates a protection against your cargo that you're trying to deliver. And so people use different things to combine these different classes of lipids into one specific lipid structure. You can use periplastic pumps. These have low reproducibility, obviously, but they can yield high volume. They do have a bit poor mixing ability, and they're not suitable for LNP precision. You can use syringe pumps. This kind of give you moderate control, but they have low volumes with limited scalability, and they have a lot of manual optimization needed. And then pressure pumps are most stable. They have a very good flow control with improved scalability and can allow bulk mixing. So these are optimal for LNP control. They can allow you to have a good size of the LNP molecule with increased encapsulation efficiency. So really, microfluidics are enabling precise, scalable, and reproducible LNP formulation that is critical for clinical translation. And then the other thing you have to think about is the lipid ratios. So as you go through different classes of lipid combination that you want to use to package your cargo, you have to decide how much ionizable lipid is needed, which hyperlipids, and how much of it is needed. And then you can perform these different validation in an early phase screening to establish which composition is giving you efficient delivery. And then as you go through your product development, you can variate the different classes to ensure that it's giving you efficient amount of lipid composition that can yield production of your mRNA in vivo. Overall, most lipid composition is going to be your ionizable lipid. This is what allows delivery of your cargo. And then you can variate the hyperlipid, cholesterol, and pegylated lipid, which is there just to enhance stability, is typically used in the smallest of quantities. It takes a lot of resources, obviously, to develop an mRNA therapeutic. And most laboratories obviously don't have all these resources to perform this type of stuff. I can say that we've had great success working with CROs such as VectorBuilder to be able to perform some of this work, synthesize, validate different classes of lipids, and essentially be able to generate clinical versus preclinical as well as clinical stage molecules that we can validate therapeutically. So you can use CROs to essentially do some of this work as well. In terms of organ specificity, most lipids, like I said, are going to end up in the liver, but people have been doing some fancy stuff to kind of tailor make them to ensure they go to specific tissues. So I've just listed a few studies here where they've been able to deliver lipid nanoparticles to the lung. People use specific peptides to penetrate the blood-brain barrier. There's some stuff that allow them to go through the spleen and the bone marrow. So there's specific things you can engineer against specific lipids to allow bypass the liver challenge and deliver it into different tissues. I will use an example of the blood-brain barrier to illustrate this point. So this is a study where they're doing a lot of screening of different lipids that can penetrate the blood-brain barrier. And then they use artificial intelligence to try and develop specific things to enhance this brain delivery system. And then they're going to validate if that's something they're accomplishing. So they take obviously the different combination of lipids, perform obviously the specific macrofluidic mixing, and then they inject it in the mice via tail vein injection to validate whether it's going to the brain. What they do is they take the pegylated lipid and they target with different molecules that are known to transport things to the brain. So you have acetylcholine, memantine, glucose. These are things that typically will go to the brain. So they tagged all these different things to the pegylated lipid and then validated whether each one of them was going to the brain more specific than another. They show quite convincingly that if they use acetylcholine tagging to the LNPs, they are able to deliver their cargo to the brain efficiently. Here you can see, let me just go to panel F over here, where if you have untargeted lipid, you can see that the intensity of delivery is low. Whereas if you add acetylcholine, you can see the intensity goes up quite significantly, and therefore comparative between the targeted and untargeted is a very good delivery post blood-brain barrier. But more importantly, they go through different exercises showing that they don't deliver it to other tissues. So to wrap it up, I'm now talk a little bit about the translational case studies. So this study obviously has been in the news a lot this week. It was covered at the annual AACR conference where they looked at a vaccine for pancreatic cancer. If you know, pancreatic cancer is a very deadly disease. Most patients, once they are diagnosed, they have about 4 to 6 months of survival. So it's a deadly disease to have. And so these guys design neoantigens where they design mRNAs containing the different mutations of pancreatic cancer. And then they use those such that they can engage with the immune system to be able to recognize those specific genetic mutations. And then they, injected pancreatic cancer patients with those neoantigens. And what they were demonstrating is that they were able to follow patients up to 3 years currently, which for pancreatic cancer, that's unheard of. Most pancreatic cancer patients, usually they live up to 6 months from day one of diagnosis of the disease. So really, this is a demonstration of a game-changing potential therapy for these particular groups of patients. I think now they are doing long-term follow-up clinical trials in advanced phases. We will see where this work goes, but this early indication is suggesting that it's really going to yield a very unique therapy for these specific patients. This is a rare disease called propionic acidemia. This is a disease driven by genetic mutations in PCCA and PCCP genes. And so Moderna designed mRNA vaccine. They designed an mRNA replacement strategy where they generated PCCA mRNA and PCCB mRNA. So if you don't know acidemia, this is a disease of multiple complications. So if you have these genetic mutations, you have neurological complications, life-threatening diseases. Patients typically are frequenting into the emergency room. So if we can deliver these two mRNAs, could we reduce these complications? So they obviously injected 16 patients with different amounts of these two different classes of mRNA. And they monitored the safety across different dosages. And then they started to monitor effectiveness of the therapy. So this is a year before patients are injected with the treatment. And the red dot basically means this is a visitation to the emergency room. So you can see that a year before injection, they are frequenting into the ER. And then they start to inject these patients around 60 days post-injection. You can see a complete reduction in the frequency of emergency room visitation. And then over time, as they continue to inject the mRNA to the patients, you can see an almost elimination of emergency room visitation, which suggests there's a very potential cure for this type of disease. So you can see here, these are different clinical trials. Obviously, initially, most of them are usually targeting liver disease, but now you can see a registration of different trials looking at cardiac ischemia, which is an extrahepatic delivery. Here you can see a trial looking at mRNA that is targeting a solid tissue tumor, so extrahepatic. There's studies looking into melanoma, suggesting that we are at a stage where we can transition to study diseases outside of the liver. So if I can conclude very quickly here, I think we've come a long way. You can see that in the exploration stage between 2002 to 2013, there was a lot of proof of concept studies, and so very few clinical trials were happening at this stage. They started to go up around 2016, and then in the 2020, 2019, there was multiple clinical trials ongoing. Obviously, these were COVID-19 clinical trials, but that was pretty much the breakthrough point where we now have a lot of clinical trials validating the potential therapeutic application of mRNA therapeutics. So key takeaways here is that there's now a strong global adoption of mRNA therapeutics, very strong validation of its versatility and scalability. So the momentum is quite right at this time for anyone to engage with mRNA therapeutic development for their specific diseases of interest. So thank you very much for listening. I hope we have a lot of time to be able to answer some of your questions.
Thank you so much, Francis, Dr. Enane. That was wonderful. A very, very thorough content for mRNA therapeutics. We have some questions. The first one is, in the pancreatic cancer trial, only 50% of patients actually responded to the vaccine. Do you think this has more to do with the patient-specific neoantigen mRNA design, or with the vector delivery system, or something else entirely?
Yeah, I think that is a classic challenge with cancer. I think the key is that they design the neoantigen based on the most frequent genetic mutations. But the challenge with cancer is like every patient might have some other things, some other genetic variations that we didn't see. Obviously, I don't know the specific reason there's a 50% response rate. But what I can say is that 50% response rate for pancreatic cancer is amazing. So I think it might be just a patient genetic variation differences that is catering to the different response rate. But I also think we are still learning a little bit more about how these neoantigens function and whether there are specific immune activators that may be better for patients, for one patient versus another. So here they were engaging T cell response, but maybe for a different patient, you may require to activate a different type of immune cell. So to be specific, I'm not sure yet, but I think it's really going to be context dependent and patient dependent.
Great, thank you. I want to remind everybody to please put your questions in the Q&A box if you have any. Um, we have somebody wondering how you are using therapeutic mRNA in your research. How much ever you can answer.
Yeah, for sure. So we engaged, so we study a lot of rare diseases and diseases where there's a one genetic mutation that drives the disease. And basically what we are trying to see is if we have patients, say, sickle cell anemia, and we know the genetic driver of sickle cell anemia, can we design an mRNA replacement strategy for sickle cell anemia that can overcome the challenges that patients with sickle cell anemia experience? There are a multitude of rare genetic diseases that we are also studying, and all of our work is really in the preclinical stage where we are evaluating a variety of protein replacement strategies and trying to effectively determine if we can treat different diseases across different organs. So there's some diseases where we're studying the effective therapeutic replacement of different proteins in the lung, in the brain. We are looking at different diseases, obviously in the bone marrow, in the context of sickle cell anemia, and also looking at potential application towards solid tissue cancers.
Thank you so much. We have another question. Which type of mRNA do you think will be the next best performer or mostly used? Linear, circular, or self-amplifying?
Yeah. I think what I can say about this question is it's a good question, right? Because not all mRNA are created equal. Um, I don't think I think it's really context dependent. If you're trying to develop a vaccine, it might be better to use a naked mRNA, not a naked, but a single-stranded mRNA. Um, I think where we need to go into is diseases where there's a chronic loss of protein. And in the chronic loss of protein, we want to try to validate these self-amplifying RNAs so that we can replace the effectiveness of this chronic loss of protein. There's a multitude of diseases where that could be a game changer for patients. So I think that all of the different structures of mRNA mean something. I don't think that one can replace the other. I think it's just dependent on what the therapeutic goal is going to be. But I think, you know, I put heavy weight on self-amplifying mRNA in the context of chronic diseases where the protein is just not expressed. And when that protein is not expressed, patients are experiencing crazy complications. I think if we can substantiate this self-amplifying RNA to therapeutic application, they could be a game changer. What we have to be careful about that is also if you too much of everything can be poisonous too, right? So what we have to ensure is how much self-amplifying RNA can we have such that it doesn't then lead to other adverse events.
Thank you. Something related to that is, have you considered using other RNAs such as saRNA or circRNA for your therapeutic purpose?
Yeah. So it depends, right? So if we have genes that are amplified, and we want to reduce those amplified genes, then siRNA becomes perfect because what we focus on is genes where there's a mutation that affects the expression of your protein. So that's when we focus on protein replacement. But for cancer and different other diseases, usually you have amplification of certain genes. So for example, if there's an oncogene that drives the tumor growth, you can develop an siRNA strategy against the oncogene and then be able to deliver that siRNA using lipid nanoparticle. What I can guarantee you, I mean, maybe I shouldn't guarantee, what I can say is that you actually probably will have a lot of success with siRNAs because they are easy to work with. I think the protein replacement, sometimes the proteins are huge, and then you have to figure out which specific piece of protein do you want to put in, right? So I think it's disease specific. It depends with what your protein is doing and whether you want to increase the protein or if you want to decrease the protein, and that's where saRNA may come in as a useful molecule. But people are doing that, so it's not out of picture.
Thank you. We have another question. Given how rapidly mRNA LNP technology has evolved to current clinical applications, what do you see as the most important limitation that still needs to be overcome for mRNA therapeutics?
Yeah. So for mRNA therapeutics, I think the biggest limitation is this issue of tissue tropism. How do we efficiently design our mRNA to access a specific tissue and then retain the mRNA that we've packaged into that tissue long enough to accomplish a therapeutic intent? So a lot of the lipids that we use typically will go to the spleen and the liver. Which type of engineering can we modify those lipids to then push it outside the liver? I think that is a huge hurdle that obviously we are experiencing. The other thing that is of interest is when we perform the specific tagging of specific ligands. So you can tag your lipid nanoparticles conjugated to a ligand and then allow that ligand to go to the tissue of interest. The thing that we then have to think about is adding that ligand, does it elicit any specific adverse event? There are things that we have to think through to be able to ensure that we're not bringing in some level of toxicity. But really the key major thing that will be a game changer is the ability to design lipids that can go in the body and be completely undetected, where you can come in, deliver your cargo, and the body does not have any idea that you actually did that. I think if we can do that, we'll be in a very differentiated state of mRNA therapeutics.
Wonderful. We have maybe one last question that kind of wraps up the contents again and highlights the things that you already mentioned. Just can you highlight the different mRNA modifications one more time?
Yeah, sure. So maybe let me just go back to the slide over here so that I can be a bit more specific. Oh, that was very early on, huh? Okay, maybe this slide. So the different modifications so the key is where are you modifying your mRNA, right? So you can modify the 5' end, and the way you modify the 5' end is you basically want to add different capping structures at the 5' end. Capping structure just means add a molecule that is going to block the ability for enzymes to recognize that 5' end, because the 5' end is biologically designed to be exposed to endonucleases. That is how we ensure that the body ensures that they have enough protein to perform what it needs to perform biologically. And then the body eliminates it using endonuclease recognition sites. However, if we are trying to deliver a protein and it's quickly degraded, we're not going to accomplish that biological intent. And so the addition of phosphodiester analogs can essentially allow these endonucleases to be able to not recognize the 5' end. There are other classes of capping structures that you can use, cap 0, cap 1, cap 2. This usually you do the identification of the methylation status of the first nucleotide, and then you can use the capping structure depending on the methylation status of that nucleotide. Within the body of the mRNA, you can perform methylpseudouridine replacement so basically the uridine is what makes mRNA unstable. And so you can replace these uridines using different things, whether it's thiols, pseudouridines, methylpseudouridines. All these can help you essentially reduce the ability of the mRNA to be degraded and basically enhanced stability. But also what we've learned is that these modifications also allow the immunity to be unable to detect your mRNA and degrade it. The poly-A tail is just modifying different sequences of the polyadenylation. I think most people use about 100 poly-A nucleotides. That's become pretty standard. But some, depending on your mRNA sequence, you can variate the poly-A tail to a specific length and try to establish which length is giving you a efficient translation of your mRNA. Obviously, I don't know if this answers the specific question that was being asked, but just those are the different modifications that a typical mRNA will undergo.
Wonderful. Thank you. And in the interest of time, I think we will wrap our Q&A right here. Thank you so much, everybody, for being a part of this gene delivery seminar series. And thank you so much, Dr. Enane, for a wonderful presentation. We learned a lot. And we hope to see you again next month. Thank you very much.
Thank you. Thank you for having me, and thank you everyone for joining.